باتاو سيندروم، داون سيندروم and إدوارد سيندروم وغيرها من المتلازمات الوراثية التي يستطيع ضيفنا اليوم تشخيصها. حقق إنجازات ونجاحات باهرة وحصل على جوائز عالمية وهو في الثلاثينات من عمره فقط. ويرافقنا اليوم أيضا صديق الدكتور بلال عزب، ضيفه وصديقه من جامعة هارفارد في أمريكا، الدكتور سامي عمر. مرحبا دكتور. مرحبا، الحمد لله على السلامة. دكتور، ممكن تحكي لنا عن الجينيتكس إن ميديسن بشكل عام؟
طبعا فالجينيتكس بالكلينيكال ميديسن is a very broad topic وفيها مجالات جدا واسعة. الأهمية الأكبر هي الآن للدايجنوستيك كلينيكال جينيتكس، واللي هو بيتكون في نوعين. في عندك الجيرم لاين اللي بتخصص بالفاميليال تايبس اوف mutations and diseases، مثلا زي الكارديوميوباثيز والهيرنج loss، لما تكون مثلا في recessive disorder that is segregating in a family. وعندك طبعا السوماتيك which is dealing with being able to take a FFPE tissue from a cancer tumor or something else and being able to identify the type of mutations in EGFR or KRAS so that you're able to help with manage therapeutics for the cancer patients. And so those are the major two lines of diagnostic clinical molecular genetics that we do in the lab, in clinical laboratories.
طيب دكتور بلال، ممكن تحكي لنا عن الاكسبيرينس تبعك والجينيتكس بشكل عام؟
Yeah, of course. أنا درست أول شيء أنا والدكتور سامي مع بعض بنفس الجامعة، وكان هو السينيور علي بسنة، فهي finished earlier وراح على هارفارد عمل فيلوشيب. و later on I joined الجامعة الأردنية، و it happened إنه اجتمعنا مع بعض مرة تانية على بروجكت هو دراسة أمراض الشبكية من ناحية جينيتكس. فأنا I went على هارفارد مع عند الدكتور سامي لأنه هو الدايركتور اوف كلينيكال جينيتكس هناك، وعملنا أناليسيز للجينز اللي بيسببوا هاي الميوتيشن. ف his facility is involved in sequencing DNA samples for patients من many different genetic diseases. وبعد هيك next plan هي we published our article last month, و there's several articles راح يطلعوا soon. وبعد هيك we're planning to understand إذا هذا هو disease causing gene إيش potential possibility إنه to package the normal gene in an adeno-associated virus، وبعدين بالتعاون مع الophthalmologist trying to rescue the disease by injecting a retina of the patients this virus. وهذا هو الجين ثيرابي. الجين ثيرابي nowadays بأمريكا هو بالretinal diseases is approved for certain diseases زي Leber congenital amaurosis for clinical treatment لأنه it passed the clinical trials several months ago. And it's very expensive. يعني one eye, you know Dr. Sam, one eye حاطين بالliterature $1 million to inject one eye بالفيروس. احنا we want to do it for يعني almost for free لهدول patients من خلال funding مدعومين for those research topics. فالبداية كانت هي من عند دكتور سامي. We're trying هلا to meet again and try to build on that.
So just to kind of continue on what Dr. Bilal is talking about. So the exciting thing about being able to look at retinitis pigmentosa patients is that the eye is one of the few sites that gene therapy is possible because it's accessible by the way that you are able to deliver that type of plasmid or construct to be able to deliver the correct gene to replace, for example, whatever mutation is causing the RP. It gives you an idea how gene therapy can be used in other parts of the body. So when you're thinking about gene therapy, you have to think about, it's not the issue, we understand what the genetic mutations are. We understand it whether it's in retinitis pigmentosa, we understand it if it's for hearing loss, we understand it if it's for cardiomyopathy, we understand it for the different systems, but the question is, how are you going to actually deliver the gene, the corrected gene to that site that requires the gene therapy. In the case of the eye, it's easy. In the case of the ear, for example, hearing loss, it's also easy. It's accessible. But for example, for cardiomyopathy, it becomes an issue. How are you going to inject a viral vector or any kind of vector? Make sure that it targets the tissue of interest and not affect other bodily tissues. At the same time, not have side effects that we knows about and some that we don't know about, and this is kind of the tricky thing. So what you guys are doing here in the cell therapy clinic, what Dr. Bilal is doing and what his team is doing is exciting because it is actually one of those things that is, going to be paving the way of how gene therapy will be implemented in the future. The reality is, in my opinion, it's still 5-10 years away before this is part of clinical medicine. But the first step is, of course, the diagnostic piece. And I think that very soon, definitely in the US and I think very soon in Jordan, everybody will be sequenced. So you have that information, you have the diagnostic profile for individuals, and then you can follow up with specific diseases in the future. And I think that it's going to be a combination. You can't have one without the other. And that's what's exciting about the germline aspect of diagnostic testing.
I want to just add على شيء حكاه الدكتور سامي يعني emphasize على point هي الثيرابي part is doable. What is really challenging هو delivery and targeting. If you're able to access tissue of interest اللي هو زي ما حكى الدكتور سامي, if it's it's doable. يعني أنا من تجربتي كنا trying to understand بالanimals. I used to inject المايس بالblood circulation بالtherapeutic virus, والproblem إنه الvirus was not able to reach to the target tissue لأنه the liver captures the viruses, it filters the viruses, so most of the viruses are taken by the liver. غير هيك كمان بالblood circulation بالsystem you have مشكلة الimmune system, it's firing لأنه there is an immune reaction, the T cells are boosting up trying to fight the virus. But as he said, I'm trying to build on what he said, إنه if you access tissues that are بسموهم immune privileged, where you can inject the retina of the eye directly, bypass the immune organs and systems, or other parts of the respiratory system, or the hearing system.
To keep in mind, Dr. Bilal spent a lot of time working on viral vectors, but he also looked at, I remember during your PhD, you looked at also nanoparticles, protein bubbles, or some other kind of mechanism. So the issue is not, again, the genetics, it's actually the delivery that's going to be interesting. It's an engineering problem more than it is a biological problem, in my opinion. And maybe viruses is the key, but it might be something else. It might be something synthetic, a synthetic molecule. And that's why, the next breakthrough will be the delivery mechanism rather than the diagnostic perspective. Diagnostic, everybody now has a large sequencing machine. In the US, you can do a whole genome sequencing in under $2,000. We're close to the $1,000 genome, and that's pretty exciting. So I imagine, in the next 5 years, everybody for large health systems and maybe, internationally, will have sequence information about will have, their genome fully sequenced and available for them for the physicians to follow up on. The next question is, what can we do with that data?
شكرا دكتور سامي وشكرا دكتور بلال، عن جد دمرتوا البرنامج. وإشي صغير من برنامج دوبامين، تفضل. إشي صغير ليبقى ذكرى دكتور سامي.
لا تطلبي مني حساب حياتي، إن الحديث يطول يا مولاتي. كل العصور أنا بها فكأنما عمري ملايين من السنوات.
It's very interesting. It's not like a job. It's not like thing you're just interesting at.
Absolutely, and to add to that, every case I feel that I work. So there's different paths in genetics that you can take. One is, for example, you're studying to be MDs. There's a clinical genetics path. What a lot of my colleagues do, they finish an MD, and then they do an either internal medicine or a pediatrics fellowship, and then after that they follow up with a clinical genetics fellowship. That's one way to do it. This is for individuals who want to see patients. Now I have another path that They work in a clinical hospital. They interact with patients who have genetic disease. They get referrals from different other clinics, from different specialty clinics. For example, somebody who works in cardiomyopathy or a cardiac clinic would either send it to the clinical genetics or they themselves would send it out to test it. After specialty, exactly. Hearing loss, we see a lot of hearing loss patients get referred to especially pediatrics actually. A lot of pediatrics eventually get transferred because there's a family history, for example, or it's a syndromic thing that we know has to have a genetic cause. That's one path that a lot of MDs take. Another path that I took, I'm a PhD, so I trained with Dr. Bilal at Medical College of Virginia. I did a 5-year PhD and then I did a 2-year clinical molecular genetics fellowship. And after that 2-year fellowship, you take a test to become boarded in molecular genetics or cytogenetics, depending on what your fellowship was. And essentially, you're boarded by the American board to be able to run a clinical laboratory, to sign out clinical cases. And a lot of MDs also pursue that. They want to work in the laboratory. They're interested in being able to run a clinical lab, or at least be able to interpret genetic data that comes out. So a lot of them still keep their clinical position. They enjoy interacting with the patients, but they also, when they get the reports, they want to be able to interpret the results themselves. So they do that extra molecular genetics, clinical molecular genetics fellowship. What I found was a lot of MDs, that was the most enriching experience, to be able to work in the clinic, but also to be able to do something in genetic lab. And they're not looking to run a clinical laboratory, they just want to be able to interact with that clinical laboratory a little bit more efficiency. And back to your point is that it's an exciting field. I honestly feel that every case that we receive, especially now that we're in the exome and genome era, every case that we receive is like a new research project. It opens new questions. It allows us there's a discovery opportunity to discover genes, and Dr. Bilal has had very interesting findings in the RP cohort that he's worked on. And I mean, it's actually fascinating that the kind of information that we're able to get out of it. Now, one of the biggest problems that we're also facing in the field is that we identify a lot of mutations and variants that we actually don't understand. Variants of uncertain significance, we call them. And that's going to be the next big thing, being able to understand those variants of uncertain significance and what they mean to the patient and to healthy individuals in terms of disease causation, or potentially all they are is just variation, normal variation across different humans. So this is going to be the next big hurdle in diagnostic genetic testing. So it's very exciting, it's very interesting, definitely keeps us on our toes.
But I want to follow up on what Sami was saying. Dr. Sami كان يحكي عن the training. So as a clinician, you can do your residency, specialty in whatever field you want, but then you can build a subspecialty, a fellowship in clinical genetics. That's one route. Another route, you can do a whole residency in clinical genetics. You finish your medical school. There is this 4-year program. Part of it, mainly pediatrics, but focused on genetic
or
disease.
Actually, they combine OBGYN and genetic. Depends on a couple of different things. If an OBGYN or pediatrics is normally 3 years in the US, you add an extra year for genetics. So it's definitely
So nowadays, there's many options for medical students to specialize in different fields, وهذول الفيلدز يكون لهم علاقة بالgenetic diseases as well. The other thing is that, as Dr. Sami said, في many mutations that are being described in literature, but the significance for those mutations and being benign or pathogenic is still uncertain. Now, while we were doing the collaboration مع بعض, we were advantaged إنه عندنا families بالجordanian population, extended families. So those variants of uncertain significance عمالهم بيظهروا معنا in those families, and we are able to classify them as benign or pathogenic, like آخر project بعتنا, right? So, and that's something is essential to report to the literature. As a clinician, if you find a mutation in the family, you need to know it. Is it disease causing, or is it just variation in the sequence without clinical consequences? So.
Okay, thank you, Dr. Sami, Dr. Bilal. It was really very nice to meet you. Thank you.